Genomics is really the study of the A, T's, C's, and G's that make up our DNA. We also do a lot of RNA-seq, so it's really the sequences that make up all the RNAs that are floating around in all our cells, providing the instructions for our cells and ultimately our bodies to do what they do. And when things go poorly in a disease, we can work hopefully to sequence all those A, T, C's, and G's, make sense of them, find out what may be causing a disease.
I think the biggest thing for people to understand is we don't treat you as a number. We really treat you as a colleague, even if it's a very routine type of process. And it's really people first.
We are heavily involved in the research. We're heavily involved in the science itself.
We have a holistic approach, so we help from the design of the experiments, from the sequencing through the data analysis. And what that means is we store the data, and we're talking terabytes of data, determine what mutations might be present, make sense of that in a way that the data will hit home and you don't have to have a degree in computer science to make sense of it yourself.
Most universities perform a very surface level analysis and just hand them over really big data sets that they have to then go through and find meaning. The U of R goes a step further than other universities.
We find to increase the value of what we offer is to be able to integrate that analysis with everything that we do. The main value of us is being able to identify what the needs are of the investigator, identify experimentation, and really come up with a strategy or solution that will help fit their needs.
Cutting edge technology is really important to us. We try to listen to all of the researchers, the type of questions they're asking, and make sure that what we're providing them is going to help answer those. We have a lot of automation that helps with liquid handling and library preparations. And then we also have some of the more technical spatial transcriptomics equipment. And then, of course, our sequencers from Illumina. We have the MiSeq, the NextSeq, and the NovaSeq here for sequencing.
One of the most recent purchases was our NovaSeq sequencer, which is a state-of-the-art, cutting-edge DNA sequencer that allows us to do human genome work for a fraction of the cost that it was years ago.
We're now sequencing whole genomes in days. It's, that advancement is phenomenal. But that has also led us to being able to look at single cell, the expression of a gene expression at the single cell level, which allows us to understand the heterogeneity of a population. And that heterogeneity can help us differentiate between healthy and disease states and help us understand the pathways that we can perturb to potentially find a cure, find a treatment.
The key value that we bring is really in the technology, how we apply it, and the results that we're generating for folks, but also helping them understand it. The people are driving all of those aspects. A lot of investigators keep coming back to us largely because of the feeling that they get working with the folks here.
You have to be passionate about finding the answers, which is why we went into science.
We're all part of U of R. We're all trying to do the same mission, and it's the quality that matters.